Welcome back after the first hands-on session for this training school. I would like to welcome for this session for the in vitro versus in vivo studies our first speaker, Paolo Bigini from the Negri Institute.
Thanks. Good afternoon to everybody. Before starting, I must confess I'm a pharmacologist, and so I'll start my talk talking about pharmacology. Why pharmacology? I think everybody knows which is the path for the approval of drugs. It consists of different steps before the approval of the compounds. But maybe it's not so well known that almost all compounds stop their path in the first phase of the clinical trial. What is this clinical trial? This is the phase 1, and is related on the safety and dosage studies for passing to the next step. What does it mean? The proper dose means the kinetics, so the study, the evaluation of the interaction of drugs, nanodrugs, and so on, in terms of half-life in bloodstream, accumulation in organs, and possible clearance. The safety is related to the follow-up of patients, of the subjects in terms of organ function and the overall health status. Why I started my talk focusing on pharmacology? Because even if completely different, we strongly believe that the approach to understand the side effects of drug nanocarriers in the phase 1 clinical trials is not different from the investigation of the toxicity of nanomaterial. The philosophy is the same. So it's the same, but when we move from pharma to tox, the situation became more and more complex because subjects are exposed to different material. The entry, the administration, in this case exposure, differ from different parts, so nasal, oral, dermal, and so on. There are acute, chronic, and permanent exposure at the same time in the same subject. And when you have to determine the overall effect, the cohort is not homogeneous. There are healthy and sick person, child, adult, old, and male and female. In order to try to be to create a more clear understanding on how it is possible to evaluate this effect, we can divide the fate. We can divide the different passage of the fate of different nanomaterial, drug, and so on. In the following parts. So, the first, the interaction with the bloodstream on biological fluids. We have to know the permanence and stability of nanomaterial. Afterward, the filter organ penetration and accumulation. The biological barrier passage is a very important point, in particular for vital organ and very, very vulnerable tissues. And at the end, but not so but very relevant, the penetration inside the cells. In all these situations, we have at the same time to understand possible clearance, excretion, and biodegradation of material. In clinical studies, phase 1, it's obvious that the subject are human. Is it possible to somehow utilize the same approach in nanotoxic studies? Quite not, because yes, it would be direct, a quick impact, more understandable the effect, but there are obviously, first of all, the most important, ethical limitation. As I told you before, subjects are not exposed to a single material, so it's not possible to create or organize a trial following the toxicity of a single material, and there is a great heterogeneity in cohorts. So, a possible alternative, now I'm back to the topic of the talk, is to use alternative models that can summarize all the steps I showed before, so excretion, accumulation, cell penetration, and in just to evaluate a single or more than a single material in terms of toxicity. It is important to keep in mind that the study of animals can be carried out, but we have to consider a maximal reduction of the number of animals used, and this is another important point, a refinement of these methods in order to reduce the sufferance of animals and also to try to perform experiments more overlappable with studies in humans. By the way, despite of this great emphasis on reducing the number of animals, if you check mouse and toxicology in PubMed, you will find that in the last 50 years, the number of studies published using these two words hugely increased. So it's true that we must reduce the number of animals, but the information still needs, unfortunately, the use of animals. Just a few words, in our institute, our approach is to try to reduce and to mimic as much as possible the human studies, exploiting, we can say, a platform that can collect different models, starting from 2D, 3D cells, organ on chip, the nematodes, and the use of animals, both healthy and diseased animals, and use a platform of tools to perform and to obtain data, and then to collect and to have some results. First of all, I want to show here very, very briefly an example of a paradigmatical study. Here, a very, very simple endpoint. We wanted to know if the metal nanofibers could impact on liver health status. To make them detectable, we level this particle, these nanofibers, and first we enrolled the animal by evaluating some parameters of health, and we divided in two groups, treated and treated. This is a correct way to start the study. Then we treated for 3 times weekly nanofibers, fluorescent nanofiber in the tail vein of mice, and at the end of the experiment, animals were sacrificed. For the whole duration of the study, a veterinary doctor checked the health status of animals like in a phase 1 study in human clinical trials. Afterwards, it was possible to exploit the fluorescence of nanofibers by optical imaging associated with X-ray to find the presence of the particle inside the body in living animals. Very, very briefly, then, we perform MRI at different time points in order to see if the overall organ, the gross anatomy of animal, didn't change during the treatment. At the same time, we collect a drop of blood to perform classical hematochemical studies, such as transaminases and so on, and to measure the hemochrome, so white and blood cells, also the hematocrit. At the end, animals were sacrificed. We separate and collect cells in order to see by fax the population, and we also perform histopathology. Just to conclude this short summary, we found that in terms of overall stress and overall status health, no difference were found among the two groups. The veterinary were blinded, so they didn't know at which group the animal belonged. Then by MRI, so in vivo imaging that can be used also, in particular MRI also in human follow-up, we found no morphological, not morphometric, anatomical alteration, but a progressive hepatic accumulation of fluorescence, so of the nanofibers, and a mild clearance. Afterwards, the blood analysis showed that in the hepatic marker of liver functionalities did not change, but a light and transient hemochrome alteration occurred. And at the end, looking at by microscopy the section of liver and by fax, we found that no necrosis of fibrosis activation was observed, but there was an increase of myeloid and immune cell infiltrates. So, just to summarize, we found that there wasn't, in this condition, a real pathological situation, clinical situation, but some important changes in terms of homeostasis, in particular of immune cells, occurred. Just to conclude my part of the talk, then I leave the floor to Luisa. There is a third important point in the 3R rules, that is the replacement. So the use of alternative methods compatible with the objective of the research project. So Luisa will explain this part of our study.
So Paolo is now Luisa. Yes. Diomede, I didn't announce you before, so also from a colleague yes. from Paolo, also from the Mario Negri yes. Institute, is continuing with the presentation. Thank you, Paolo.
Yes, good afternoon to everybody. And when we talk of replacement, taking into account the third R of recommended by EU, we, in general, think to the use of alternative animals, alternative strategies that can replace the use of animals. But instead, yes, I prefer to talk about the use of complementary methods, of complementary animal models. Among the different complementary animal models that we can use in pharmacology, but also in nanotoxicology, today I'd like to talk to you about the use of C. elegans. That is the animal model that we selected in our group to do nanotoxicology studies. So we can imagine C. elegans as an intermediate model which can offer relevant information that cannot be obtained in vitro using 2D, 3D organoids or organ-on-chip, and that can be very informative to perform future experiments in vertebrate, recalling the concept of also refinement, for example. And C. elegans, for those of you who are not confident with this animal is a little nematode, invertebrate nematode, that is usually living in the soil and waters of temperate zone of the planet. It's completely transparent, and so it's very useful, for example, when you want to determine the biodistribution or accumulation of nanomaterials, particularly if they are bound to some fluorescence dye. It eats bacteria, is non-pathogenic and non-parasitic. But why C. elegans is so interesting for toxicology studies? First of all, it is, although it is quite simple from an anatomical point of view, it is formed by specialized cells. So there are different cells that sometimes have also counterpart invertebrates, and the number of cells is constant. It's always constant because C. elegans are almost hermaphrodites, so the number of cells composing the animal are constant. The 60% of C. elegans genes are homologous to humans, and also the 40% of proteins are preserved in vertebrates. And in addition, among the genes that are homologous to humans, we have also some genes responsible for human diseases. And this is not trivial because, particularly in the field of toxicology, is important not only to determine the toxic effect on healthy individual, but we well know that fragile subjects, so subjects with a disease, are those that can be more affected by the side effects of drugs or compounds. Also, the protein homeostasis involves biochemical and molecular pathways that are shared, conserved in mammals. And C. elegans has a shorter lifespan. It lives for a maximum of 3 weeks. And so it is also easily and also easy to determine is in a not expensive way the effect of a toxicant on the whole lifespan, including also the aging phase of the life. It is a very inexpensive model and is devoid of legislative restriction, and it represents also a good system for high throughput screening. This is the reason why C. elegans can be used for determining or predicting the toxicity in vertebrates. In fact, it can provide relevant data on the effect of a toxicant on the metabolism, on the digestive tract, on the reproduction, sensory and neuromuscular systems. In literature, there are many examples of modes of action of toxicant that act in with similar mechanism. in C. elegans and vertebrate. And interestingly, the IC50 value determined in this nematode are predictive of the LD50 in rodents. So it's possible to use C. elegans to select the possible range of doses of concentration that you can then test in vertebrates. And in addition, C. elegans is a nematode, and nematodes are the most abundant animals on Earth. As I told you before, nematodes and C. elegans too is very widely present on the heart, particularly in the subarctic regions, and they play a fundamental role in the transformation of organic micronutrients and in control of the population in aquatic and terrestrial microorganisms. So they are good indicators of the quality of soil and of water. This is the reason why C. elegans is also used for studies of environmental toxicology. And as you well know, the possible release in the environment of nanomaterials, advanced nanomaterials, is particular of interest in the last years. And for all this reason, C. elegans represents a good model also for this type of studies. In fact, some standardized tests for evaluating the soil and water, the water quality, are already available. And but what kind of study, what kind of toxicity studies can we perform using this nematode? We can perform studies evaluating some phenotypic behavior, so that included the life of death, so the lifespan and the span, the feeding behavior, the motility, reproduction, larval growth, but we can also perform biochemical and molecular studies to dissect the mechanism by which a toxicant can exert its toxicity. And this is particularly interesting because sometimes the use of vertebrates is so expensive and so difficult, at least in the first phases when you considered an emerging or a new compound that can be toxic, or a new nanomaterial that could be toxic, but for which there are no sufficient information. So I think that I finish the presentation, and just before to conclude, I would like to thank you for your attention, and so if there are questions, I don't know what happens, but... Paolo and I will stay here until Thursday, so if you have any question or curiosity, feel free to come to us.
Perfect, thank you, Luisa. And yes. So next up we have Dario Greco from Tampere University.
Yeah, here. All right, good afternoon, everyone, and thanks so much for the kind introduction and having me here today. I will bring you in the next 20 minutes through the journey that I did when I entered the nanosafety field now about 12 years ago, when things were still simpler and we were just speaking about toxicology, and how I had to change the way how I see the business as new concepts were coming in, and now we are interested in understanding sustainability in a broader sense. I will speak about mechanistic toxicology, so essentially how we explain the steps that occur during an exposure and how we model the response of biological systems to this exposure. Now, the nice thing is that mechanistic toxicology fundamentally is chemical agnostic. It means that we can reuse the same thinking and the same modeling approach to virtually any of the chemicals that are out there. And this is just to give us the scale of where we are at the moment. The chemical universe we are thinking today exploded in the last couple of decades. And we have the same problem. So Paolo was approaching this from the pharmacological point of view, and indeed we have exactly the same problem in the way, kind of a specular problem, right? So we are producing new chemicals at a faster pace than we could ever test carefully their possible adverse effects, and at the same time we are not developing enough new drugs to take care of all the big challenges of modern society. So how do we balance these? Well, in traditional toxicology, essentially we have been thinking in terms of testing the effects of one chemical at a time. And the focus has been essentially on observing apical effects, so really phenotypic effects, in order to derive the safe doses from the less safe doses. But it's obvious that this is not enough. It's way too expensive, it has also ethical implications, and most of all, it doesn't inform us on what happens in the biological system during the process. And if we learn this, probably we can also start developing better chemicals. So it's clear that toxicology faced a couple of decades ago already a big shift. So from just describing the endpoint effects of an exposure towards building models that could. Is it better now? Okay, sorry guys. Thank you, Steffi. So better models that could explain actually what happens in between, so all the intermediate steps. And that's where a new, in a way, complementary approach kicks in, which I like to call a systems toxicology, where we not only get information on the apical final endpoints of the exposure, but we also describe the mechanistic steps that occur during the process. Now, if you're a molecular scientist like me as a background, then this essentially means looking into the biological systems during the exposure and understanding how their molecular setup is affected at different doses and different time points. So that's where toxicogenomics comes in, which is fundamentally based on the use of omics technologies, which give you a powerful opportunity to get a snapshot of virtually all the molecules of a certain type into your system. Now, in this image, I have stacked different levels of investigation you can do with these technologies in a specific order. So what is important to understand is that not all the molecules in the cells work the same way. Some of them respond really fast, but then once you remove the exposure, they will go back to the steady state. So they're really good at capturing acute responses. Now, the more you go down into this stack, the more you get into the investigation of data layers that can help you to understand what remains altered, even after a long time, you eventually remove the exposure. So, which is actually what we face in real life, right? So how many times we open the news and we read that someone died because drinking a huge amount of a certain chemical? Very rarely, fortunately. But what we observe is that being exposed continuously to nano doses of something eventually will change your susceptibility towards getting a new disease. So this is the kind of challenge that toxicology has had in the last 10, 15 years. What is also interesting to understand is that toxicology in this fashion becomes a complementary discipline to epidemiology. So in toxicology, we try to test things in order to predict what would happen if a long-term global exposure would occur. While in epidemiology, we do exactly the same thing, just that we look retrospectively at what has happened when the exposure has already occurred in the past. And yeah, so as I was saying, the idea is that toxicological tests have been designed to capture acute responses, while the long-term response to chemical exposure has been traditionally more a topic of investigation for the epidemiology. So the idea really is to understand, like, how can we reuse this information in order to learn better the long-term effects in a toxicological setting. So what we have to understand is that whenever we observe in real life a biological system which has lived a normal life, and we look at the molecular build-up, well, we see a mixture of different signatures of past exposures that have occurred and probably are not happening anymore in the present. So imagine the possibility of getting a totally unexposed biological system for the first time. Then you would start with kind of a basal amount of molecules in the system. Then you get a certain chemical, there is an acute response, toxicological testing captures these, but then you remove the exposure and some signature remains there to be observed in long terms. Then maybe in this situation, a new exposure comes in, again, acute effects that, again, a toxicological test has been designed to get, but then at the steady state, you see kind of this mixture of situations. So now we are left with a mess, because how do we reconstruct the exposure history of a biological system? So here we have to first thing acknowledge the fact that we have to be able to build systems and models that can capture things in a dynamic range, so during the time. Another thing that we have learned is that chemicals behave like the paintings by Monet. So essentially, not two lilies look the same in his paintings. And that's exactly what we observe in toxicology. So each chemical gives exactly a very specific signature that is not similar to anything else. And now for those who are more active in the nano safety, they have learned that nominally the same nanomaterial in the same biological system will give a completely different response, maybe due to different dispersion protocols and so on and so forth. So we are now left with a big puzzle and a really difficult situation where we would like to start observing groups of responses. The point here is that we have used traditionally to use even molecular investigation in order to generate lists of molecules that are altered by an exposure. However, lists of molecules remain lists of molecules, and this doesn't tell us much. But then we know that, for instance, all our cells are equipped with the same genes, just that they work in different combinations with each other, and these different interactions between the molecules determine specific functions that eventually bring to different anatomical formations and different functions, and now organs and tissues that interact with each other characterize individuals, and individuals of multiple species interacting with each other builds the whole planet Earth. So now you understand that why not we would use a theoretical framework, which is systems theory, that allows us to forget for a moment whether we are speaking about molecules, tissues, individuals, or species interacting with each other, and we look at what I call the fabric of life. Now, the great advantage of systems theory is that you are applying exactly the same mathematical modeling thinking to whatever you are analyzing. So you're focusing on the interactions much more than the objects that interact with each other. Now, in an attempt to model the molecular responses in multiple species in consequence to nanomaterial exposure, so we started from the analysis of over 150 different nanomaterials in multiple biological systems, so in vitro, in vivo, multiple species. And when we looked at bluntly the list of genes that are associated to each one of these, we saw again one nanomaterial, one signature situation. So there was no way to reconstruct a common way to respond by biological systems. However, when we started to investigate the way how the genes interact with each other and how they are co-regulated together in different biological systems, we hit the jackpot because we landed on a number of a few transcription factors, members of a zinc finger family, which would be consistently present in the promoter regions of genes that respond to nanomaterial exposure in multiple species. So this, de facto, was the very first One Health or planetary health molecular model that would explain how multiple species on this planet behave in consequence to nanomaterial exposure. The funny thing is that when you look at the whole business in systems theory terms, not only molecules, individual species interact, but even data can interact. So this will be a topic tomorrow, I believe. But what you can do is to start collecting enormous amount of data in a network fashion. We call these knowledge graphs, where the nodes are data points, and then the connections kind of are a meaning of something happening in between different types of data. And for instance, in our lab, we have now built a knowledge graph that is quite comprehensive, over 70 million data points, and still growing. And now the funny thing is that humans are quite kind of allergic to these kind of huge network structures, but fortunately artificial intelligence is quite at ease with the analysis. So things that you can do with the knowledge graph include, for instance, finding new connections between, let's say, a chemical and a disease, which is not necessarily described in the data, but might be implied while looking at the pattern of connectivity between the different data points. So this is a great kind of way to learn new things. And for instance, we use this approach to kind of clarify what are the intermediate functional steps that occur during the COVID-19 infection. So we started from a set of genes that we know interact with COVID-19 virus, and we knew which kind of genes were differentially expressed, so altered in function at the very last stage of the COVID-19 disease. And then by using this large network structure, we clarified some of the intermediate steps that occur during the infection at different stages. And we could use these to find new drug targets that might not only inhibit the entry of the virus or the final immune response, but also multiple intermediate steps in the infection. What I'm trying to convince you here today is the fact that in order to do this game successfully, we have to think in terms of space-time. And now, the problem is that you know, for instance, from the beautiful images that are brought back by the astronomical telescopes we sent out there in the space, that it doesn't really make sense to read these kind of images by concluding, for instance, that these two galaxies are closer than these two galaxies, right? Because we know that the light that is captured by our telescopes is actually coming from totally different time scales, right? So some of the light has been emitted by the stars much long time before, and some come from structures that have evolved much later in the life of the universe. And that's exactly what we need to do also to fully understand all the steps in multi-scale that occur in chemical biological interactions. Because, for instance, when we look at only strictly to toxicological events, the interactions that chemicals have with biomolecules happens in a matter of nano to milliseconds, right? So really fast. But then the effects that we see on tissues and cells might occur between days and months. And then eventually we need decades or even multiple generations to appreciate changes that concern the whole environment. That's why we have a lot of problems to make political decisions at this level, because these effects are so slow that our politicians are stupid enough not to see them, right? So that's where we are, right? So we have a completely different space and time scale at different dimensions in the toxicological space. Now, I'm sure that this audience is quite acquainted with the idea of adverse outcome pathways. They are emerging, not anymore emerging, quite established actually nowadays, modeling frameworks that consist of chains of key events that explain a causative relationship between different things that might happen in multiple scales, so molecular, cellular, systemic, and so on, that link an exposure to an adverse outcome. And interestingly enough, when you look at the whole AOP network, they indeed form a network. So again, we have nodes, and again, we have connections to study. And so by doing this kind of analysis in the beginning of the COVID pandemic, we could find a striking association or a striking similarity between what was known at that time to be the AOP for acute respiratory disease due to COVID-19 infection and an AOP that many of the people in this room are very familiar with, which brings to pulmonary fibrosis from particulate exposure. And through this analysis, we could predict that pulmonary fibrosis could be a long-term effect of SARS-CoV-2 infection, which at that time we couldn't observe epidemiologically, but that unfortunately, or fortunately, scientific terms, we could observe later on in the real population. So this is really a powerful way to kind of model the steps, the multiple steps that occur, and eventually predict the future. Now, when you look at the effect of chemicals in this mechanistic fashion, all of a sudden, different chemicals start grouping, and now the grouping is not only driven by their intrinsic characteristics, or how structurally similar they are, but also by what kind of chain of mechanistic effects they trigger in the biological systems that are exposed to them. And of course, like when we speak in terms of in vitro and vivo extrapolation, then the big question is how much we can learn from an in vitro system about the real thing, if we admit that animals are the real thing, and this is not always so much for granted. So we developed a new way to look at toxicogenomics responses, and which is based on AOP, systematic AOP annotation of the molecular alterations that we observed after a certain chemical exposure. And we proved that indeed this can be really useful to capture things in vitro that recapitulate what happens in vivo. So this is kind of a nice, interesting tool that we can use for in vitro and vivo extrapolation. More than that, we have also observed, interestingly, that the AOP framework can also be used to define in vitro models applicability domain in the same fashion as we have learned this morning about QSAR modeling. Essentially, you have to ask yourself, what are the genes and the molecules that a certain biological system in vitro or in vivo is equipped with to start with? And then you will get a nice map of what are the AOPs or the mechanistic effects that a certain biological system can observe. And so in vitro, in vivo extrapolation, then we are interested in observing chains of events that are overlapping between the in vitro and in vivo system, while there are some mechanistic aspects that can be captured only by an in vitro model or eventually by an in vivo model. So this is like a straightaway way to give a nice confidence on what an in vitro system can observe. Now, when we go beyond toxicology, we know that what we have discussed so far has also social and economic implications before going to the whole planet and eventually the environment. So how do we put this whole puzzle together? Well, in one of the projects that are actually living in this school this year, which is Insight, we are now expanding the concept of AOPs to include also social and economic chain of events that can explain mechanistically what are the social and economic impacts of the chemicals. And they are actually related to each other. So let's say that you observe that the production of a certain chemical will trigger a cascade of events, maybe going through child labor, which will bring a certain community to have lower socioeconomical status. Now, we know that in a community where there is lower socioeconomical status, lifestyle changes in a way that probably we start eating bad, we start smoking, we start doing a number of things that eventually will increase the inflammation in our tissues, for instance, the lung. And so this can be kind of nicely modeled as a modifying factor that triggers the adverse outcome pathway that brings to lung cancer. At the same time, for instance, a chemical that is released in the environment which triggers a cascade of events which brings to decreased biodiversity, we know has an indirect impact on sensitization and allergy. So we know that in environments where we have less species around us, our kids get more often asthma and other types of allergies. So you see now, it starts building kind of a coherent cause-effect mechanistic framework that we can use to explain different moments in the life cycle of our chemicals. Nicely enough, there are multiple types of computational modeling techniques that can tackle down specific questions in different scales. So in molecular, we have clearly QSER, we have docking, we have learned today. We have, at the biological level, toxicogenomics data, which can be used to derive internal dose and point of departure, and so on and so forth. So we can actually build an ensemble of models that can address different scales and then build the whole system together. So in this exercise that Angela, Sarah, and our group, together with the colleagues from Athens, are doing, we have put together PBK analysis with toxicogenomics. So for long-term exposure, we have defined specific point of departures or kind of internal doses that might trigger a biological effect. And then this results in a nice reconstruction of an adverse outcome pathway that explains how a liver function is, mechanistically, how liver function is impaired from long-term exposure of this type. Now to conclude, where I believe we are at the moment. So in traditional toxicological testing, we were just scratching the surface of the system. And at this level of observation, where we stop at observing how many animals are dying or how many animals are developing a disease, here we don't see much of overlap between how different chemicals behave. And so it becomes extremely difficult to implement safe and sustainable by design because we don't know what kind of characteristics we should consider. However, the more we dive deep into the mechanistic understanding of what is happening, especially when we consider the multi-scale, so from molecular to the whole planet health level, understanding how these interactions in multi-scale happen, then eventually we start observing behavior of chemicals that are overlapping until we eventually reach this that I consider the golden area where we can derive information that can be used to understand One Health, so multiple impacts at the planetary level as well as SSPD. All right, I'll stop here, and I'd like to thank, of course, the crowd of people that I proudly work with up in the north in Finland, and of course the projects we are involved in and our funders. And I would like to say at this stage that we are really heavily active in recruiting. So if you have heard anything today that was tickling your curiosity, please have a chat with me in the next days. Otherwise, thanks so much for your attention.
Thank you very much.
Here is Amaia Garcia from GAIKER. She will talk to us about the risk assessment part in SSBD.
Okay, good morning. I will try to speed up a little bit. So the title of my presentation is Safe and Sustainable by Design and Risk Assessment Approaches in Composites Development. So what we tried to do here was use a European project, which is the Repoxible, use the case studies we have there to explain a little bit about what are these approaches. So at first, I will go briefly through the implementation of safe and sustainable by design, and then I will talk about what type of materials do we have in the European project, and then I will go directly to the case studies. And with that, we will talk about the approach, the recycle, and the risk assessments. Okay, so briefly, this was also explained yesterday by Iranzu. This figure is from the European Environmental Agency, but yesterday we talked about the chemical assessment, and it's very similar. So basically what we have to do is follow different steps. The first one would be identifying the product we want to develop. So if we focus on function instead of the form, we can develop different candidates like in a more easier way to consider them as sustainable. Because, for example, if we want to prolong a life of a product, we need to increase the durability and the reparability. So if we develop a less complex product, we can separate it in components, for example, and we can repair them or reuse them, so we can decrease the resource use, and we can foster the circular economy. So that's the first step. Then we have to think about the impacts that this product will have, and then we have to see it along the life cycle of the product. So we have to see what chemicals do we have, are they hazard or not, and what volumes do we use. We have to see the resources, where they come from, and what resources do we use to produce this. We have to see the energy consumption, so how much energy do we consume to produce this, and what impact will have in the nature. Then the third one will be considering the sustainability of the product we want to develop. So there we have a life cycle assessment, and here the most important thing is to have good quality data. And then last, we will have to do a balance between safety considerations and sustainability, because we can have a product that is super safe, but we used a lot of energy to produce it, so it's not sustainable. Or the other side, it can be sustainable but have hazard chemicals, so there has to be a balance there. I will go through this because we already know. And okay, so within the Repoxible project, we used this kind of material. So just in case someone don't know what we're talking about, epoxy resins are a family of synthetic resins that contain different parts, but they need to be cross-linked with what is called curing agent or hardener. And on the other side, this simple mixture doesn't really provide the properties we want, so we have to add more things, more additives, and then we form what is called the composite. So in the Repoxible case studies, we used two sectors, the aerospace sector and the automotive. So both of them have quite differences in what they require. For example, the aerospace needs high technical requirements, and the automobile needs high sustainability requirements. So what there was at the moment when we decide to use these case studies, well, both of them need to reduce the use of resources and emissions. And they both need materials that are lightweight, that are recyclable, and they become circular models. And what we have, or what there was at that moment around the epoxies, was that they were weakened when we were using in specific conditions, like high temperatures, for example. In those conditions, they released toxic fumes and microparticles, and there is a low possibility of recyclability or reutilization. So the plan was to produce high-plane, high-speed, use our composites to create this high-plane, high-speed aircraft. In the case of aerospace and automotive, the fuel cell low-weight car. So each has its own requirements. We propose requirements based on materials and processes, but also based on sustainability and, well, the functionalities that we want to put in the materials. So the approach of the Repoxible, according to the SCCBD, was to develop a bio-based epoxy composites that are recyclable, sustainable, and cost and energy efficient. while keeping high performance during use. So it follows an holistic approach across the entire life cycle, okay, from the raw materials and production to the use and the end of life of the composites. How we applied the SSCBD? Well, we divided in 3 steps, let's say. The first one is in a lab scale where we select the raw materials we're going to use, and we do a kind of proof of concept. So we run a very simplified SSCBD assessment, and we will focus mainly on the step 1, which is the hazard characterization, and also in the steps 2 and 4, but very, very simplified. Then we scale up the process. We focus on the step 2, which is the worker exposure and the manufacturing and recycling processes, so we take in mind that, and we perform an early life cycle assessment. And then finally, we run the full SSCBD assessment with a full life cycle assessment using these both case studies. So the implementation of the SSCBD approach brings in-depth knowledge in the whole material life cycle. This is very similar to what I just explained, so I will run through it. So here is the composite system we use in the Repoxyable. We have the epoxy resins that are added. There is a hardener added, so we have the pre-peg, then we cure it, and then we finally add more additives and have the composite, as I explained before. But all this process was done considering all the life cycle of the product. So the way that the epoxy resin was prepared was using this system called DCLE, the polymerization closed-loop epoxy resin, which I will explain later, but it increases sustainability because it allows us to have a more recyclable material. In case of the curing, we used infrared curing, which decreases the energy consumption and increases sustainability of the final product. And the additives that were added were also considering these requirements, that lowering the energy consumption and increasing sustainability. So all this was done before starting the production. Sorry. The requirements decide. We define which were the requirements, and then according to that, we selected the materials. Then doing this DCLE resin system, we synthesized the polymers, we did the formulation, and then we added fibers or whatever, and had the design of the composite. In all these steps, the material safety assessment has to be done, and also the recyclability of the thermostats and different components also has to be done. So from the composite that I previously showed, you have this recycling process where the composite is depolymerized, and we have different components separated. So with filtration and other different steps, we can obtain again the resin back so we can reuse it. So the requirement list included 6 lines of evidence, which was performance, technological, safety and sustainable, legal and economical. So these were the 6 lines that we were focused on. Then we prioritized with this requirement the product solution based on the application that we want. Then we did the inventory of the materials and the material safety assessment and this SSCBD. And then finally we selected the materials where we are doing that. So which are the requirements? As I said before, performance, technological, safety, sustainability, legal, and economical. Well, these were the requirements, and specifically what we needed was, for example, in terms of performance, the material needed to be resistant at 300 degrees during 1 hour of flying, for example. It needed to have a lightweight. We needed between 50 and 80% of bio-based content. So these were the data that we selected from those requirements. And in this picture, the orange line represents what we have now, and the blue one is what we want to reach. So it would be like improving what we have. Okay, so we have the inventory of the materials. We select the benchmarks because they are the materials we want to compare with what we are producing. We want to see how they can compare with what is already in the market. We identify the information needed for safety and different sources of information. And we create a kind of Excel file where we fill in all the data that we are getting. So this is an example of an Excel table. We have on one side the performance requirements of the aerospace, and on the other side the performance requirements of the automotive, and the safety and sustainability, which goes for both. In terms of the human health hazards, well, there's a list of the safety indicators, and what we have to do is something that we are already doing in this hands-on training. So we have to identify all the materials that we are inventorying, if they are already characterized with this information or not. So we classify them according to this category 1, 2, 3, 4. The category 1 will be the most toxic. If there's no information, in this case, we put like a yellow miss test. And if there's no needed, well, we put it in green. So this is the results we had from the safety assessment. We this is for the aerospace case study. So here we have the benchmark materials, and then from there we have the materials that have been developed in the reproxable. So as you can see, the information we have, there are some information found, but there are many yellow boxes, which is information that is missing. So in order to fill those gaps, we selected specific in vitro assays, both for human and environmental. And they were done between GAIKER and University of Ljubljana. And with this data, we can fill these gaps that we have in the table. So that's the real life, but well, that's the ideal life, but in the real life, we have many issues going on. The first one would be the product that we have to test is not soluble, or it doesn't disperse good, so we're using in vitro methods that we need to try to think about how to use this problem, because we need to expose the cells to the product, and if it's not soluble, we need to find out ways to do the test. We can find many data gaps that are not possible to fill, so how are we going to treat them? We have to take into account that, as Alberto explained yesterday, some of the assays might be really expensive and they might be time-consuming, so we have to take into account that. And it happens also that depending on the jurisdiction, the risk thresholds are different. So we have to use like a universal standard criteria. Finally, this is something that is going on in this project. We are doing this life cycle thinking, which in general terms is like consider the whole picture before deciding what is truly sustainable. So I know you cannot see it, but this is like a questionnaire that it has to be filled while we are advancing with the product results. So from the very beginning, we will have a few information, but then this information will grow until we can have the whole life cycle assessment. And finally, just 3 take-home messages. Like safe and sustainable by design is not just a single step. You have to see it as a whole. The holistic approach ensures that what we create today doesn't become a problem tomorrow, so we have to take into account many, many things before doing it. And then as a researchers, designers and decision makers, we have the opportunity, and I would say the responsibility, to integrate safety, sustainable, and circularity from the very beginning. So thank you.